Good morning, dear children of class 12 bio. Today we are looking at the starting of the fifth video of the 8th chapter of Evidences and Theory of Biological Evolution, and we will be looking at part 5. Last time we looked at the evidences of morphology and anatomy, and today we will look at the evidences from embryology. Now, what is embryology? Embryology is the study of development of an organism from egg to adult. A comparative study of the development shows that the embryos of different vertebrates, they resemble more closely than their adults. We see in the diagram on page 305 that the embryos of fish, salamander, turtle, bird, dog or human, at the same stage they resemble so closely that it is difficult to distinguish between them. Such resemblance of structures is a clear proof of evolution and indicates that such forms are evolved from common ancestor. Some points of evolutionary evidence are: the embryos of all vertebrates, they develop notochord and gill clefts. Notochord is partially or fully replaced by vertebral column in adults. Similarly, the second point of evolutionary evidence we can look at is that the development of certain organs like heart, brain, and ear in vertebrates indicates their common ancestry. The heart, during its development in birds and mammals, passes through two-chambered and three-chambered stages before becoming four-chambered later on. Thus, birds and mammals have originated or evolved from fishes having two-chambered heart. Now, the third evidence we see here in embryology is temporary embryonic structures. The gill clefts are temporarily embryonic structure. Let us look at this diagram. In this diagram, we see the young tadpole of frog, and we see that the gill clefts are temporary embryonic structures which develop in the embryos of all land vertebrates and are absent in their adults. Gill clefts are useful in fishes that live in water but are useless in land invertebrates. Notochord is developed in the embryos of all vertebrates, but it is replaced by vertebral column in adults. We see in recapitulation theory that Ernest Haeckel postulated in 1866. His theory is very famous. It states that ontogeny recapitulates phylogeny. I would repeat, dear students, recapitulation theory of Ernest Haeckel states that ontogeny recapitulates phylogeny. Now, what is an ontogeny? Let us take the meanings one by one. Ontogeny means embryonic development of an organism. And phylogeny, what does it mean? Phylogeny means the evolutionary history of a race of any organism. So, recapitulation means the repetition. So, this theory, which states that ontogeny recapitulates phylogeny, can be understood in these easy words. This theory means that embryo in their development repeat the evolutionary history of their ancestors. Again, let us look at this example of figure 18 on page 305. Let us take the example of a tadpole of a frog. The tadpole of a frog is fish-like, as we see here, due to the presence of compressed tail with tail fin and gill slits and a lateral line system of sense organs which are fish-like organs. These fish-like features in development of frog, which is an amphibian, clearly indicates that its evolution is from fish-like ancestors. Recapitulation theory also applies to plants. Let us see an example. Example in Protonema, an early stage in the development of moss and fern. The gametophyte resembles the filamentous green algae. It resembles the filamentous green algae in structure, physiology, and growth pattern. This suggests that algal ancestry is there of bryophytes, which are mosses, and pteridophytes, which are ferns. Another scientist, D. Bayer, in 1828 suggested that organs achieve complexity from simpler forms due to their needs. As needs arise, so more complex structures arise from simpler structures. Now let us take these structures one by one. Let us look at the first structure. The general characters appear before special characters during organism development. From more general, less general, and finally the special characters appear. The third point D. Bayer stated was that an animal during development departs slowly and continuously, that means progressively, from the form of other animals. And the fourth point which he made was that young stages of animals are like young or embryonic stages of lower animals, but not like adults of those animals. So hereby we have explained about the evidences from embryology. In our next video, we will continue with the evidences from cell biology and biochemistry. Wish you all the best and take good care of yourselves.